I'm Daniel Levine, and this is the Bio Report. Vaccitech first gained attention as co-inventor of the COVID-19 vaccine licensed to AstraZeneca. The University of Oxford spinout is developing a pipeline of T-cell immunotherapies to treat and cure chronic infectious diseases, autoimmune diseases, and cancers. The company's lead experimental therapy is a potentially curative monotherapy for chronic hepatitis B viral infection. We spoke to Bill Enright, CEO of Vaccitech, about the company's platform technologies, how they work, and how they enable the development of more effective immunotherapies. Bill, thanks for joining us.
Oh, thanks for having me. I appreciate it.
Listeners may be familiar with Vaccitech from the work it did with AstraZeneca to bring a COVID-19 vaccine to market. We're going to talk about Vaccitech's pipeline of T-cell immunotherapeutics to treat and cure chronic infectious diseases, autoimmune diseases, and cancer. How does a vaccine differ from an immunotherapeutic, if at all?
No, thanks. So typically you think of a vaccine as being protective. So it generated an antibody response, which protects or prevents the body from getting infected with a bacteria or a virus or something like that. And the immunotherapeutics actually are treative. So you're treating diseases after you already have them. So chronic infectious diseases like we're involved in with hepatitis B or HPV infections, or in the event of cancer, in mutated cells.
In terms of identifying antigens to train the immune system on, is the approach the same for a vaccine versus a therapeutic?
Yes, it's very similar. You're trying to maintain, uniqueness, right? So you're trying to get antigens that the body will recognize, the immune system will recognize, and that's very similar. Now, there's slight differences, and if you're developing a vaccine, you want antibody responses, and so the antigens may be more geared towards the antibodies. And in the event of the therapeutics that we're working on, you're going after T-cell epitopes. So there may be some little variances as a result of that, but for the most part, we try and use, full-length proteins.
Vaccitech is not only pursuing infectious diseases and cancer, but autoimmune diseases as well. In an autoimmune disease, I think of the challenge being quieting down the immune system rather than revving it up. How would an immunotherapy for an autoimmune disease work?
So you're revving up a different part of the immune system. So in the event of when you're looking at cancer or in chronic infectious diseases, you're trying to ramp up the CD8 positive T cells. So that's the part of the immune system often called killer T cells. They recognize the infected or mutated cells and destroy them. In immune tolerance, or what you're trying to do is ramp up the Tregs, and that's part of the T cell system that dampens the immune system.
Once you've identified one or more antigens to properly activate the immune system, use mRNA to code for these and deliver them through two vectors. You're using a modified chimpanzee adenovirus for a prime treatment and then boost it with a modified vaccine, Ankara vector, for booster doses. Why do you use different vectors for the prime and the booster?
Yeah, so many people may be familiar with this from the COVID-19 work that was done, and what they showed during that, and actually our scientific co-founders have showed for the last 20 years they've been working on this, is that if you use different platforms, you actually are able to stimulate slightly different immune responses. And so we come in with the chimp adenovirus and deliver that prime, and then come back with the second virus, the pox virus or MVA, and deliver the boost. And our scientific co-founders at the University of Oxford, Adrian Hill and Sarah Gilbert, really spent about 20 years looking at this and looking at different combinations of different viruses platforms, of mRNA platforms and DNA platforms, protein platforms, and different combinations. And they really tested this empirically in phase 1 clinical studies and showed that this combination of different viral platforms, the adenovirus first, followed by the MVA, gives you the highest magnitude and longest duration T cell response.
You also have a separate platform technology you call SnapVax. What does this technology do, and how do you use it?
So the SnapVax was a really clever technology developed by some outstanding chemists at Johns Hopkins University and the NIH in Bob Cedars' lab. And it's really designed to mimic the adenovirus, although synthetically. So instead of a live virus, they're doing this through chemistry. And they did a great job in developing this system. It's a self-assembling peptide nanoparticle technology that allows you to incorporate the antigens of interest, but also link it with immunomodulators. So if you're going after cancer indications, you can use immunomodulators like TLR7/8 agonists, which help to stimulate the CD8 positive T cells that I was mentioning. And if you're going after immune tolerance indications, you can add things like taurins or rapamycin, which help to stimulate the Tregs that I was talking about. So it's very complementary to our existing technology platform. You can use it in combination or alone.
Given the broad potential of the technology, how do you go about prioritizing the indications you'll pursue?
Yeah, so we're going after significant areas of unmet medical need, right? So the indications in hepatitis B, there are more than 300 million people chronically infected around the world with hepatitis B, almost 300 million women infected with human papillomavirus. So significant market opportunities here where we have the potential to treat large numbers of patients, and where we think T cells are going to be important to their cure. So that's one of the key things as well.
Let's talk a little more about hepatitis B. How does the condition manifest itself and progress?
Right. So hepatitis B is often called the silent killer. So many people don't even know they have it for long periods of time. So and many people actually cure the disease naturally. So about 90 to 95% people that get infected with hepatitis B, their body takes care of it, never even knew they had it. It's 5 to 10% of people that get chronically infected with hepatitis B that sometimes go on to develop further complications. So as I mentioned, almost 300 million or over 300 million people chronically infected. About almost 1 million people die every year from cirrhosis and cancer of the liver. So it usually manifests itself through, very similar symptoms to flu, actually. So you get fever, abdominal pains, things like that, that end up bringing you to a physician to look at.
We've seen tremendous success in treating hepatitis C and even curing it. What does the hepatitis B landscape look like, and does this pose greater challenges to treat?
Well, I think the hepatitis C market was a fantastic win for the pharma industry, and showing that we can really, bring a cure to a very tough disease. And hepatitis B is much like that. And I think many in the field will say that it may be even a little bit tougher to cure, and that it also will require a multifaceted approach. So we're looking at a number of different ways to try and go after this, but a lot of progress is being made right now.
Is your expectation that your therapy would be used in combination with other therapies?
There is that potential. So we're evaluating VTB300, our product candidate for a potential functional cure in hepatitis B, either alone or in combination with other therapies. So, typically right now, patients, when they're diagnosed as having a chronic HBV infection, they get put on nucleosides or nucleotides, so basically daily antivirals that they have to take. It doesn't cure the disease normally. It's a very low cure rate, maybe 1% to 2%. So we're using it in combination with that. But a functional cure is really defined as trying to get people off of those nukes, get them off their antivirals for a period of at least 6 months. And so we're looking at combining with things like checkpoint inhibitors, because we know that in HBV and chronic HBV patients, they have T cell exhaustion, very similar to what happens in oncology. And in fact, these checkpoint inhibitors were actually discovered in infectious disease and not oncology. And so we can use really low doses of the checkpoint inhibitor, so you don't have the same side effects that you see in the oncology indications. And we're also looking at other things that will potentially disrupt the viral replication, such as siRNAs. And those folks like Arbutus and others have been working on, and they've gotten very good responses in decreasing the surface antigen, which is the biomarker of the hepatitis B activity. And the problem is once you stop giving those therapies, that surface antigen level starts bouncing back. And what we've seen with VTP-300 is with a single dose, we're able to decrease the surface antigen levels for up to 9 months with a single injection. So it's the only molecule that we're aware of where you're able to see that sustained reduction in surface antigen.
You mentioned your lead experimental therapy, VTP-300. What is it and how does it work?
So again, it's using this prime boost approach. So we take the major proteins of the HBV virus and use those as antigens. So they're incorporated into both the adenoviral vector as well as the MVA vector, and then you treat as a prime boost. And again, trying to stimulate the body's immune system to recognize these antigens. And we're using that in combination with the checkpoint inhibitors that I mentioned, as well as siRNAs and various combinations to try and see if we can provide a functional cure here.
What's known about it from studies that have been conducted to date?
So far, the data look promising. So we released data at AASLD last fall, and we'll have additional data at EASL in June, and with our first in-man study. And as I mentioned, so this is the first immunotherapy that actually showed a reduction in surface antigen. So as a monotherapy, we got about a half-log reduction in surface antigen. In combination with low-dose checkpoint inhibitor, we got almost a log reduction in surface antigen. And again, as I mentioned, the key thing here is those reductions in the patients that responded. Those reductions lasted out to 9 months, which is the last time point we tested.
And what's the development path forward?
So we're still trying to get to show functional cure. As I said, what we'd like to do is to be able to take those patients that responded and have no detectable levels of surface antigen and take them off their nucleosides to see whether they stay down. And so that wasn't part of the original clinical trial, but we've got 2 ongoing phase 2 clinical trials where that is part of the protocol, and we hope to demonstrate that. So we'll have some interim data from both of those phase 2 clinical studies later this year, and we'll have additional data from those studies sometime middle of the next year.
Vaccitech has had the benefit of royalties and milestone payments from AstraZeneca from its COVID-19 vaccine, but your R&D costs are rising as demand for the vaccine wanes. How far will existing cash take you, and what's the plan for funding the development of your?
Right. So we in our 10Q that we announced last week, we had $191 million of cash, and that takes us almost through the first quarter of 2025, and that's funding. We've got 9 active programs, including the Vaxzevria, the licensed vaccine. So we do continue to get royalties from that, although much reduced. So we will continue to push these programs forward aggressively, and hopefully be able to utilize key milestones over the next 2 years to then do some additional fundraising to continue to move these promising programs forward.
Bill Enright, CEO of Vaxitech. Bill, thanks so much for your time today.
I really appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.